Thank you for that introduction, Luck. And thank you all for making it here today to hear me talk about a series of studies that I put together over the course of my PhD on the invasion of Centuria stoibi across northeastern United States. And I was just chatting with Luck about the nomenclature or the taxonomy of this plant, and it's a mess. But the common name is spotted knapweed, and so I'll be using spotted knapweed over the duration of this talk. And so I'm sure a lot of us are familiar with spotted knapweed. It's this invasive plant that's very problematic in parts of not Western America or Northern or Western America in general, and even in Western Canada too. And so spotted knapweed has been invasive in Western North America for a long time, where it has had devastating effects on agriculture and on land. It's native, spotted knapweed is native to Central Europe, where it came into North America in the late 1800s. The first couple of data that we have on the invasion of spotted knapweed or the introduction of spotted knapweed is actually here in BC and in New York. However, these are not the only introductions of spotted knapweed. There have been a couple of other introductions that we've seen from genetic analysis, but these are just the first couple of populations that we've seen that was established, and it was mistakenly introduced as a contaminant of alfalfa seedlings. Spotted knapweed is present in a lot of provinces in Canada, but it's not necessarily invasive in all of these places. So we know that it's problematic now in BC. It's invasive in BC, but it's not necessarily invasive in other provinces in Canada. And the same goes for the United States. So spotted knapweed is present in almost all states in the United States except two states. I think that's Oklahoma and Georgia. And so the parts of the United States where spotted knapweed is determined to be invasive are typically in the northwestern part of the country. And so that axis of Idaho, Washington, Oregon, Montana are really problem areas for spotted knapweed, and they've been problem areas for a very long time. In the upper Midwestern part of the country, Michigan and Wisconsin, to be more specific, spotted knapweed is also a big problem and has been, it's invasive, and there have been a lot of studies on spotted knapweed invasion in actually those parts of the country. However, in the northeastern part of the country, where a lot of my talk is going to be concentrated on, there have not been a lot of studies on spotted knapweed, and the notion was that this part of the country is not conducive for spotted knapweed. However, from herbarium records, we've been able to reconstruct the spread of spotted knapweed over time in the northeastern part of the United States, and we've seen that over time, the amount of the locations in which we are seeing spotted knapweed has actually been increasing. So in 1935, there were just a few locations. However, by 2000, there have been a lot of locations where spotted knapweed is present in the northeastern USA. And I actually have a more recent map that shows that there's been a lot more locations where spotted knapweed is present in the northeastern part of the country. However, there has not been any studies that have been looking at why spotted knapweed is invasive in that part of the country. And so it's an emerging invader in the northeast, and it has increased in population drastically over the past 10 to 15 years. And so my study was interested in looking at the environmental factors that explain the presence and the density of spotted knapweed at different scales within northeastern USA. I constructed a model to do that, and based on the results of that model, I conducted an experiment to look at how spotted knapweed responds to some of these environmental factors in the field. And using the results of my experiment, I built a population demographic model to understand how the population growth rate of spotted knapweed changes with environmental conditions, again, based on my field experiments. Okay, so the first question. In order to answer the first question, I built species distribution models of spotted knapweed. And so what are species distribution models? These are correlative models that the idea behind these models is to estimate the potential distribution of an organism based on the current distribution and based on environmental factors. And so essentially, you are saying the probability of the occurrence of an organism is a function of the environmental conditions. And typically, in order to build these models, you would get occurrence records, which are basically presence-absence data, coordinate data. You would get some environmental locations based on those presence-absence data. You would choose an algorithm, and there are many different algorithms that have been used, ranging from generalized linear models to machine learning approaches. And based on the results of your model, you would develop a series of predictions of the probability of your organism being present in your area of interest. And so in order to get these occurrence databases, I trawled many different, in order to get the occurrence data rather, I trawled many different databases, a global database, but also more regional databases for New England. But because I was also interested in looking at the influence of density, and I'll talk about why I was interested in density in a bit, we went out into the field and we collected data on densities of spotted knapweed. And so this was us going out into the field in different parts of New York State. And so this was us in upstate New York in the Adirondacks, and this was on Long Island, New York. And we chose these two different locations to look at this, to look at densities, because these are ecologically different areas. So Long Island is very suburban. There's high population density. It's quite, there's a lot of disturbance. On the other hand, the Adirondacks is this preserve, so there's not, a lot of people there, and so there's lower disturbance compared to Long Island. And so I got presences of spotted knapweed across the northeastern United States. But in addition to that, I also got densities of spotted knapweed in the Adirondacks and on Long Island. And so typically when building distribution models, people have used presence-absence data. So they use coordinate data to build the models. However, I was interested in looking at density because I was wondering that when you collect data on presence-absence, all you get is the organism is present in this location. There is no indication of the success of that population. There is no indication of whether that population is failing, whether it's just one plant or 20 plants. So if it's one plant, if it's 20 plants, you get the same kind of data, right? And so with density, I was interested in seeing how that might influence the predictions that we get from the model. So I wanted to know is that data enough? Is the presence-absence data enough to tell you all you need to know to predict the probability of your species of interest being in the location you're interested in? Or is the additional data that you get using density, which of course you have to put in more effort to get density data, and would that additional effort be beneficial in making the predictions of your model better or not? Would it tell you anything different from the presence-absence data? And so I had presences for Northeastern USA, but I also had density data in Adirondacks and on Long Island. And so basically, these are some of the points in which we got density data in Adirondacks and on Long Island, in the Adirondacks and on Long Island rather. And we saw that basically spotted knapweed occurs at different densities. Some are very high, so the larger circles represent higher densities. And so we had some high density, we had some low densities of spotted knapweed in both locations. And so using all of this data, I used presence-absence data to build the distribution models for the Northeast, for Long Island, and in the Adirondacks. But in addition to that, I also used density to build distribution models for Long Island and for the Adirondacks. And the additional information that I thought density would give us would be, given that we know that spotted knapweed is already present in a location, what factors explain the differences in density between these areas where spotted knapweed is already present? So we know that with presence-absence data, we know that the organism is already present. But would the differences in density tell us anything different? And so for the environmental data that I wanted to use, I chose the amount of sand in the soil pH, precipitation, temperature, land cover, a categorical variable, and I used land cover as my index of whether the area was disturbed or not, and then the canopy cover. When people build distribution models, there are a couple of approaches that you might take. So some people just take 30 or more variables, they feed it to the model, and they get a result, and they allow the model to tell them what's important. Well, I was lucky enough that people had built distribution models for spotted knapweed in Europe, and I was able to extract the variables they found important and to bring that into my own modeling. And so again, this is the land cover data. And so the areas that are developed, I use that as an index of disturbance. So areas that are developed tend to be more disturbed compared to all of the other areas. And for my algorithm, I chose boosted regression trees. So this is a machine learning approach that combines regression trees, which are classical regression trees, with boosting. So classical regression trees, they've been used for a long time. And basically what they do is that they split your data into two groups based on some decisions. So in this case, the decision is whether tree cover is greater than 40%, whether it's true or false. And this is just, this is not real data. And so whether it's true or false, the data is then split. And then again, at this point, another decision is made whether sandiness is greater than 32%. And so as you go up the tree, so this is the root of the tree. As you go up the tree, the data is continually split based on decisions. And with boosted regression trees, you build a ton of these trees. You build about 1,000 of these trees. And then you combine them in order to minimize your error. And you use your combined tree to make predictions. And so some results now. Across the Northeastern USA, which again are presence-absence models, this is just occurrence data, I found that the most important variable that explains the presence of spotted knapweed is percent sand and land cover. So those are the two most important variables that explain the presence of spotted knapweed in the Northeastern USA. The amount of sand in the soil, and land cover again, which was my index of disturbance. And with regression trees, you can look at how a variable relates to the probability of presence. So on the y-axis is the probability of presence of an organism, which can range from 0 to 1. And on the x-axis is your variable of interest. And so looking at sandiness, what this is showing is that as the amount of sand in the soil increases, the probability of finding spotted knapweed increases. So spotted knapweed likes areas that have more sand in the soil. For my index of disturbance, which is land cover, I found that you had more spotted knapweed occurrences in developed areas. So areas that were developed had more spotted knapweed had more spotted knapweed data. So in the Adirondacks, which again, I had two models for the Adirondacks. I had the presence-absence model, and I also had the model using density. And so I found that for the presence model, land cover and tree cover explained the presence of spotted knapweed, while for density, tree cover and temperature explained the density of spotted knapweed. So this suggests to me that there is some additional data that you can get out of using density information, right, that the presence-absence information might not tell you. And the same goes, okay, so, and also a regression plot of tree cover in the Adirondacks, suggesting that as the tree cover increases, the probability of finding spotted knapweed decreases. So it likes open areas. And on Long Island, I found that for presence data, precipitation and land cover explain the presence of spotted knapweed, while tree cover and temperature explain the differences in densities for spotted knapweed. So in general, this suggests to me, again, so looking at the impact of precipitation in this plot, as precipitation increases, the probability of finding spotted knapweed increases up to a point. So there's an optimal point after which the probability of finding spotted knapweed decreases. And so this suggests to me that at different scales and in different locations within Northeastern USA, different factors explain the presences and the differences in density of spotted knapweed. So in the Northeast, the amount of sand in the soil and land cover explain the presence of spotted knapweed, while in the Adirondacks, land cover and tree cover explain presence, while tree cover and temperature explain density. On Long Island, precipitation and land cover explain presence, while tree cover and temperature explain the differences in density. And so there is some additional data there's some additional information that you might get if you use density in species distribution models. And again, if you, as a land manager, if you are interested in controlling spotted knapweed, Depending on the location you are, you might want to adopt different strategies. In addition, if you are interested in limiting the spread of spotted knapweed compared to if you are interested in reducing the density of spotted knapweed in an area in which it's already present, you might want to adopt different strategies because different variables explain what's going on there. So this is an example of some of the plots in which we got spotted knapweed, in which we collected count data for spotted knapweed. So this is the kind of typical canopy cover for spotted knapweed, and this is the kind of disturbance that we see for spotted knapweed. So this is an area where people drive ATVs, for example. And so again, I mentioned that land cover is a proxy for disturbance, and tree cover is a measure of the amount of sunlight reaching the plant. And these two variables were very important variables that came out of the models that I constructed. And so based on this, I wanted to conduct experiments to see how spotted knapweed responds to these variables, which people don't typically do. People just get the results out of their models, they publish it, and they forget about it. And so the question I wanted to ask in that case was, how do different life history stages of spotted knapweed respond to some of these environmental factors in an experimental situation? And to do that, I looked at phenotypic plasticity of spotted knapweed at different life history stages. And so the idea of phenotypic plasticity in general is that it is the ability of a genotype to alter phenotype when exposed to varying environmental conditions. And so if you imagine that each of these lines represents a genotype, and if you are looking at a trait for your given plant species, and this is an axis of of the environment, which can be really any environmental condition. And so the first plot here shows that the organism is not responding to the environment. So under the environmental condition, the trait remains stable. In this case, it shows that the organism is responding to the environment, but all the different genotypes respond in the same way, suggesting that there is phenotypic plasticity. And in this case, all the organisms respond to the environment, suggesting again that there is phenotypic plasticity, but they respond in different ways to the environment, suggesting that there is genetic variation for phenotypic plasticity. And so the idea of phenotypic plasticity being important in weedy plants was proposed by Herbert Baker in '69, in which he suggested that invasive plants should be tolerant and should be plastic, should be plastic, and they should be able to respond to a wide range of environmental conditions. And so he was the first person that suggested that, for invasive plants, this might be a quality or a characteristic that might help. And so I wanted to test experimentally how spotted knapweed responds to light availability, disturbance, and intraspecific density across different life history stages of the plant under field conditions. And so I set up a growth chamber experiment to look at how spotted knapweed seeds respond to light. And so in my growth chamber, I got seeds from Long Island and from the Adirondacks. I had two light conditions, full light and light filtered through a 50% shade cloth. And so I monitored the experiment for 25 days. I used failure time analysis in order to take care of sensor data. So what happens with some of the germination experiments is you end the experiment at a certain time, but it is quite possible that some germination might occur after you've ended the experiment. And so you have to censor that data, and failure time analysis, which is a type of survival analysis, helps take care of that possibility. And so I found that the germination probability of spotted knapweed was better under light compared to under the shade cloth. But I also found that there was phenotypic plasticity in response to light availability. So the mean time to germination was much higher under shade compared to under light. And in the Adirondacks, the mean time to germination was higher compared to Long Island. And this makes sense because the Adirondacks is this very shady location. So seeds that are from the Adirondacks take a longer time to germinate compared to seeds that are from Long Island. In addition to that growth chamber experiment, I wanted to look at field experiments in order to see how spotted knapweed responds to disturbance and intraspecific density. And we've seen why I chose disturbance. It came out of my models. But why density? Well, across all the different populations that I visited for spotted knapweed, there was a lot of variation in the densities of this plant. And so I wanted to see if this would, come out in my field experiment. How does density and disturbance interact in order to uh, help the success of spotted knapweed? And so I was interested in seeing how disturbance and intraspecific density influence the growth of spotted knapweed under field conditions. And I wanted to see which life history stages of spotted knapweed display greater phenotypic plasticity. And I will talk a little bit, I'll talk in a bit about the different life history stages of spotted knapweed that I explored in this experiment. But I conducted my research in the Stony Brook Research and Development Park, which is this nice place very close to my old campus that I could do research. And so in 2014, this shows how the site I used looked before the disturbance treatment, and after I imposed my disturbance treatment, this is how the site looked. And so I was trying to simulate the kind of roadside disturbance that spotted knapweed typically experiences on Long Island, which is disturbance that's due to heavy machinery and construction equipment. And so by 2015, there was already some growth. And so my experiment lasted for 3 years. By 2015, there was some growth in the disturbed plots, but the growth had not reached the levels of the undisturbed plot. I'm showing you this because I'm sure we are all worried about me going out there into the field and planting invasive species. And so I'm showing you this because I put these seeds in, I put the plants in the soil. So I germinated the plants in the greenhouse. I then took them out to the field. I transplanted them to the field. I knew where they all were in the field. I had a map of all my plants in the field, and I removed them after the field experiment. So I kept track of every plant, and I removed everything. And so there's no need to worry about me planting invaders. And so my experimental design in the field. So I had a split plot design in which I had 4 plots with disturbance and 4 plots with no disturbance. And within that, I had low and high density plots. And so these are the life history stages, these are the life history stages of spotted knapweed. So most of the reproduction of spotted knapweed is from seed. It grows to form a basal rosette, which is not flowering. It then flowers, produces these beautiful purple flowers for an invader, and then releases the seeds, which again germinate. But I was specifically interested in looking at phenotypic plasticity in the rosette and in the adult or in the flowering plant. And I was interested in looking at in dividing this into life history stages, particularly because most studies of phenotypic plasticity tend to be at earlier life history stages. And I wanted to see if there were differences in phenotypic plasticity between the earlier life history stage of spotted knapweed and in the later life history stage of spotted knapweed in response to the environmental conditions that I chose. That is intraspecific density and disturbance. And so in 2014, I planted 768 plants. By 2015, a lot died, but by 2015, I had some vegetative plants, some reproductive plants, and by 2016, I had 58 vegetative plants and 295 reproductive plants. Some of them also died by 2016 rather. And so this is the data that goes into my phenotypic plasticity analysis. And so the traits I measured in the field were mainly morphological traits. So I measured the number and the length of leaves, the number and the length of stems of reproductive plants, the number and the length of leaves of vegetative plants, and then I measured the number of flowers produced. And again, because out of an abundance of caution for invasive plants in the field, I covered all the plants with these jewelry bags so that even though they flower, they do not get pollinated, and and they wouldn't produce seeds. And so I took that caution. And so some results. In 2015, density was not important, so the plants did not respond to density. So this is leaf length, high and low density. There was no statistical response. This is leaf number for vegetative plants, high and low density, there was also no statistical response. And this is also stem height. There was no statistical response to density. And I'll talk about why that might be the case later. But for disturbance, there was a strong response to disturbance for leaf length and for stem height. There was none for leaf number. In 2016, it was also the same thing. No statistical response of any of the traits to density treatments in the field. But again, they still responded to disturbance in the field. And the reason I think this might be is that, so the way I chose the density treatment, I went out into the natural populations of spotted knapweed. I measured the average distance between adult plants, and then I used that to, I brought that into my density treatment in the field. However, it's quite possible that because I measured the distance between adult plants, I had 3 years of field experiments. The plants had not matured to the point where they would start interacting with each other. So the effect of density wouldn't be as strong yet. And I had to run my experiment for just 3 years because, I have to graduate. So, but I would expect that if you let the experiment go on for a while, then it's quite possible that the influence of density might come out at later points. So I also had survival plots for flowering probability. And so the probability of flowering was also very high for the disturbed plants, for the plants in the disturbed plots in 2015 and also in 2016. The axis here is different. In 2015, just about 10% of plants in total had flowered, while by 2016, about 40% or thereabouts flowered. But I also looked at this for density, and density again was not significant for the probability of flowering in 2015. In 2016, density was actually significant, and so it's likely that influence has begun to kind of show, and it might, if I had let the experiment go on, that might be, I might have seen a stronger influence of density in my experiment. And so I was also trying to look at the phenotypic plasticity of adult plants and of juvenile plants in response to disturbance in the field. And so I found that juvenile plants tend to be more plastic compared to adult plants. And I found this was the case in both 2015 and 2016. So the plasticity of the plants in response to disturbance in the field was higher for juveniles compared to for the adult plants that have started reproducing. And so in general, I found a strong signal of disturbance on the growth and reproduction of spotted knapweed. Juveniles tend to be more plastic than reproductive plants in response to disturbance. And it's likely that this phenotypic plasticity at the younger stage is more important, and it helps the plant to adapt to this new environmental condition. And at the older stage, this phenotypic plasticity is no longer important. Finally, I constructed demographic models in order to see how the population growth rate of spotted knapweed changes with the environmental factors that I chose in the field. And so this in general was the structure of the environmental factors. So disturbance, I had in general 4 combinations of treatments. So we have disturbance present, high density, disturbance present, low density, disturbance absent, high density, and disturbance absent, low density. And I wanted to see in which of these cases would the growth rate be highest in the field. And so in constructing my matrix population model, I used 3 life history stages. So in the plants in the seed bank can develop to being vegetative, and vegetative plants can become reproductive. And this is on a yearly basis. Reproductive plants can also transition to go into the seed bank, or they can produce seeds which become vegetative in the next year. In addition, plants in the seed bank can go back into the seed bank. Vegetative plants can remain vegetative. Reproductive plants can remain reproductive. And so So this is the population growth rate lambda, and I found that the effect of disturbance on the population growth rate was higher than the effect of density. So under low density, when disturbance was present and when disturbance was absent and when disturbance was present, the difference between the lambdas was 1.05, while under high density, the difference between the lambdas was 1.21. In addition, the influence of density was on the population growth rate was not as strong as the effect of disturbance. And so I found that disturbance increases the population growth rate more than density does. However, we would note that all of these lambdas are actually very high. And I think the reason they are very high is because this was an experimental, this was in the field, but it was semi-experimental because I put the plants in the field, I watered them for a while in order to get them to stay in the field and not die. And so those growth rates are very high and may not be typically what you would see in natural populations. However, I think what might be more important here would be how those treatments influence the population growth rate. So how does disturbance impact upon the population growth rate, and how does density impact upon the population growth rate? So I also did elasticity analysis. And so what this does is it looks at which life history stage contributes the most to the population growth rate. And I was wondering if this was going to be different for the different experimental treatments I had. However, I found that in general, the life history stage that impacts the population growth rate the most tended to be the transition from vegetative to reproductive plants. And so this was the highest for all of this, for all of the treatment conditions I had. The transition from reproductive back to vegetative was also pretty high. And so it seems that these are the two stages that are very important for the establishment of spotted knapweed, regardless of environmental condition. And so the conclusions I can draw from my experiments is that the environmental factors that explain the presence and the density of spotted knapweed differs with scale. So at different scales across the Northeast USA, different factors explain presence, but also different factors explain the density of spotted knapweed. And so if you are interested in controlling spotted knapweed, you have to know what you are trying to control. Are you trying to control its spread to new populations, or are you trying to control the spread within the population? Are you trying to decrease density, or are you trying to minimize spread to new populations? And so if you are interested in any of those, you would look at different environmental factors, at least within the Northeast USA. I also found that spotted knapweed non-reproductive plants displayed greater plasticity compared to reproductive plants in response to disturbance. And finally, I found that disturbance had a greater effect on the population growth rate than intraspecific density. And the most important transition was from juveniles to reproductive plants. Thank you. And if you, and so I was just showing this. So one way by which people try to control spotted knapweed is they train dogs to detect spotted knapweed in the field, and they go in there and they get it. So it's fun. I know. So I'll take questions now. Yeah. Oh. Yeah. So the thresholds I put in there were for illustrative purposes. So I was just trying to show how the model works, but how those thresholds are actually selected would be the model selects those thresholds. So you feed your data to the model, and it actually selects the thresholds that it thinks would minimize your error. But for this, in this case, these are illustrative, and I just put that in order to explain what the model actually does.
Right. But the models before you don't have to like figure out what it would be like yourself, like beforehand.
You don't have to. You have to specify your error. You have to specify how many trees you have. You have to specify the boosting approach you want to use, but you don't specify this threshold. No.
You ever come across like a lack of data in all the different databases you're pulling from? And is there an opportunity possibly for like a citizen database or spotted map that does?
So yeah, so the data that goes into those databases are essentially they are citizen science data from when you pick up I don't know iNaturalist and you say this is I found spotted knapweed in this location and it's all filtered to those databases. We did find a lack of regional data and that's why we went into the more kind of regional databases. So one of those databases was specific to Pennsylvania and one of the others was specific to I think New York or thereabouts. And so um, was there a lack of data in general? I'm not sure if there was a lack of data in general or spotted knapweed is just not in those locations. I think it might be that. So if I look at the, so right, so if I look at this map, for example, um, in general, there's not a lot of data out of Maine and Vermont for spotted knapweed, and I think the reason why that might be is because it's just not doing very well in those locations well yet. So is there an opportunity for a database for spotted knapweed? I think so. I think it might be we might be getting to the point where that might be necessary because um, it has shown no signs of decreasing. There have been numerous kinds of efforts, so both biocontrol efforts, physical efforts to trying to control it, and that's not been successful. So that might be one thing to do.
So interesting talk and interesting require more components of land size and density.
Thank you.
Snapweed, snapweed had to get there, and we had it dispersed across the last 20 years or so. So, did you have any thought on that dispersal strategy? What components are successful in the plant and how it actually moves into those new locations?
So there are two major ways by which spotted knapweed disperses. So there's the natural dispersal, which is mostly short distance. So it releases the seeds from the capitula when that dries up, and that's very short distance. The more long distance dispersal, and I think this is how spotted knapweed was spread to various areas in New York, is by heavy machinery. So trucks that go collect sand in an area and they go dump it somewhere else, or people that are walking, trucks that are walking along the roadsides, for on construction and things like that. And so those are the more long distance dispersal strategies. Well, not strategy, but that's what helps spotted knapweed to disperse to those new locations. And so, yeah, so I think a lot of the dispersal for spotted knapweed to the new locations that we were seeing was actually due to the long distance strategy by construction equipment. Yeah.
Do we know if napkins are designed for both like are napkins? So could you say that your results are probably relevant to napkins in Alaska or
So there have been some genetic analysis that suggests that the knapweeds that we see in different parts of the continent come from different populations in Europe. As to whether, in constructing my models, I actually trained my models with knapweed data from across the continent and used that to predict what's going to possibly happen in Northeastern USA. So I would, and so I would say that I don't necessarily think that knapweeds are going to behave the same way in different locations because there's also an environmental component to it. So the environmental conditions in those new locations might be important to show how knapweed is going to succeed in those locations. But the actual plant might the response of the actual plant to those environmental conditions again will depend on the environmental conditions, and I can't really speak to what's going to happen in locations in which the environmental conditions are not representative in my model.
In germination between two genotypes, Adirondack and Iowa.
Yeah.
And I was wondering if you'd looked at other morphological or physiological traits between those two genotypes. And then a follow-up question is your field study, right, that was done on Long Island.
Yes.
Are the populations on natural invasive populations of spotted knapweed more dense on Long Island than they are in the Adirondacks? And if so, if you use the Adirondack genotype in your field study, perhaps you would see different results from density.
Yes, so the for the field experiment, so what I used were all populations for Long Island, and I don't necessarily expect to see. So I had done some pilot studies before I did my field experiment using populations from Long Island, from the Adirondacks, and from other parts of the United States, and the results were not different in the pilot study, and so I did not expect that I was going to see different responses at least within the first couple of years. However, when the plants start interacting, when they get dense, I might see different responses in the way the plant, because the populations on Long Island actually are more dense than the populations in the Adirondacks, and so I expected that that density would contribute to how the plants are going to respond to density under field conditions. But I did not have the opportunity to test that because my Adirondack populations did not survive on Long Island, did not survive enough for me to have an experiment on Long Island. Yeah. The first question, I don't think I answered that.
Oh, so other traits, morphological or physiological at different life stages rather than germination between the two.
Yeah, so I looked at just morphological traits, but it's very possible that what I'm seeing on the morphological level might be the physiological traits might be contributing to that, and so it's very possible that I might see different responses physiologically compared to the responses I'm seeing morphologically, and so I didn't look at that, but that's very possible.
Variables that you had in the during
Yes.
which way were they going?
So
They're asking you limiting things.
right, so I showed precipitation. So what happened with temperature and precipitation was actually very similar. Like for all of the locations I had, what happened was actually very similar to this. So they would start out by not doing very well, and then they would reach a threshold temperature, a threshold precipitation where they would do very well, and then it would then drop.
And the minimum temperature?
It's minimum temperature during the growing season, and so that was for me, that was from April to October. Right.
And did your data have like a wide range of that, or like
Yeah, so the range was pretty wide. Yeah.
enough to see that?
Yes, enough to see that peak. Yeah.
Last tricky question. You ever go back to your field site?
Oh yeah, no, I was actually very worried about that because I was actually the first person to do an experiment on that field site, and so anything that happens can be traced back to me. So no, I did go back several times to see if I had spread napweed, and I hadn't, and I hopefully that stays. So I did not actually allow the plants to seed in those sites, and so it's very unlikely that anything's going to germinate there.
Oh, that's right.
Oh boy, I hope not.